Madam Amy will be presenting on the changing landscape of forensic taphonomy in UK education and practice. So kindly, Madam Amy, please tell us as you have 20 minutes to discuss with us. Thank you very much.
Thank you so much for that lovely introduction. Hoping you can see me, and I hope you can also see my screen, but I'm not sure at the moment. Is anybody able to confirm if you're able to see my screen?
Yes, so it's
wonderful. Thank
Yes,
you very
Amy,
much.
we can see you. Amy, we can see you. So you can
And can you see my slides?
yes.
Okay, wonderful. Yeah, good morning, afternoon, evening, I guess, depending on where you are in the world today. And thank you so much for the organising team for inviting me to present at today's events. So my name is Amy Rattenbury. I am a senior lecturer in forensic science at Wrexham University, which is in North Wales in the United Kingdom. I'm really pleased to speak to so many colleagues today who are committed to advancing forensic science practice globally. And to be part of this conference that is highlighting some of the important research-informed education and professional development that we're seeing across various forensic disciplines. Today I'm going to talk to you about how forensic taphonomy is evolving, particularly in the UK, how education and operational practice and some of the practical research that's happening here is reshaping its future. A lot of conversations in the UK often focus on the challenges that we face, especially some of the difficulties that there have been in establishing a dedicated human decomposition facility. I want to try and move away from that a little bit today and shift the narrative. I want to show that without human research facilities, we can do things that are exciting, meaningful, and innovative. That is already happening in our classrooms, in our labs, our fieldwork sites, and through professional collaborations. Before I get started properly, I do want to just offer a small advanced apology. I am working from home today to deliver this presentation, so there is a chance that Bella, my 10-year-old Springer Spaniel, might make an appearance. She's a retired search dog, and she's not quite accepted yet that not every online meeting is for her. So if we do have a slight disruption, I apologise. I hope you'll forgive Bella and me for that. Okay, so to frame today's discussion then, I wanted to tell you a little bit more about myself and the perspective that I bring. So after completing my undergraduate and postgraduate qualifications, I did a teaching qualification, and then I worked across several colleges and universities in the UK, teaching across forensic science programmes at various different levels. And that's where I really developed my passion for forensic taphonomy, but also for innovative teaching practices. In 2016, I joined Wrexham University, and this move allowed me to develop both of these interests. So I lead on our BSc Forensic Science and MRes Forensic Anthropology programmes, as well as supervising some postgraduate research, and I'm also doing a doctorate in education part-time alongside this. So what this allows me to do is maintain pedagogic research and operational links to practice as well. So in addition to being an academic, I work as a consultant to national and international organisations, particularly those that involve cadaver dog training, disaster victim identification, and forensic recovery operations. And it's this operational experience that I think directly informs my teaching and research. It allows me to make sure that it's grounded in real world demands, some of the ethical complexities and evolving technologies that we're seeing in contemporary forensic practice. So it's that combination then of academic research and professional operational experience that I think a lot of us are seeing now is changing this landscape in forensic taphonomy education. So I'm hoping to tell you a little bit about challenges and how we are kind of uniquely building on those in a UK context. So before we move on to the broader discussion on forensic taphonomy, I just want to talk a little bit about the university where I work and our programmes. So we're located in North Wales. We're about an hour southwest of Manchester. Historically, many people had never heard of Wrexham outside of the UK. That's improving a little bit thanks to Ryan Reynolds and Rob McElhenney and the ongoing success of Wrexham football team. And thankfully now it's much easier to introduce the university when I'm speaking at conferences and things. So we focus on being dynamic and being career focused. We want students that are not just incredibly knowledgeable, but they're also able to meet the demands of their chosen industries. And we've been lucky to be ranked first in Wales for student satisfaction and for graduate prospects in forensic science, according to the Complete Universities Guide. So this is all about how we've strengthened our curriculum and committed to make sure that what we're teaching students actually allows them to go into professional practice. A lot of that has been through investment in facilities. We have a dedicated crime scene house. We have flexible laboratory spaces, a mock courtroom, and we also have the only licensed decomposition research facility in Wales. And there are just a few others in the UK. So this really allows us to focus on the practical aspects of the subject and the things that can only really be developed by doing it, by analysing it, and by problem solving skills. And we've got a wide range of different teaching methods that we're using. One that I particularly enjoy is something called Scale Up, and I'll discuss this more later on. But the idea is that we integrate that classroom-based learning, that practical hands-on approach and research as an essential part of education, not something that is separate from it. And that's where engagement with forensic practitioners, collaborating with police forces, all of those sorts of things have become really key to us because it helps us to inform our teaching and our research. So, forensic taphonomy then. I just want to spend a few minutes defining this. It might be a term that some people are already familiar with, but we know that terminology is not synonymous across the globe. So it might be that you're aware of elements of this, but didn't necessarily know that that's what we call taphonomy. The word taphonomy comes from a Greek term taphos, which means the grave. But essentially in forensic science, it's the study of biological, chemical and environmental processes that affect human remains from the moment of death all the way through to the moment of discovery and recovery. And what's essential here is that it's a complex series of changes that occur after death. So it's not just about soft tissue decomposition, it's about disarticulation, it's about skeletonization, and sometimes even preservation or destruction of remains. And the more knowledge we have around that, the better that we can inform our forensic investigative practices. And taphonomy can help us to answer some of the really critical questions in forensic cases, such as how long has the individual been deceased? What are the environmental factors, temperature, humidity, soil chemistry, and how have they influenced the rate of the decomposition? Has the body been moved after death? Has any trauma occurred before, during, or after death? And how might environmental or even animal interaction have altered the evidence at the scene? So through this detailed observation and interpretation of the post-mortem changes, forensic taphonomists can contribute to constructing those timelines and then reconstructing the history, assessing the trauma, and hopefully as well helping to identify the deceased. What I think is really important to understand is forensic taphonomy isn't a single discipline field, and it's fundamentally multidisciplinary, and it needs to draw on expertise from several different domains. A lot of it stems out of forensic anthropology and archaeology, interpreting deposition environments and skeletal modifications. But there's forensic entomology and insects colonization, looking at biological clocks for PMI estimation. There's forensic ecology and soil science, thinking about those environmental conditions, and elements of forensic pathology too, and the post-mortem investigation process there. Increasingly, we're seeing a lot of research in forensic microbiology and also forensic botany, and we're expanding our capabilities in there. So, for example, looking at bacterial succession patterns or plant growth dynamics and how they can indicate time since death, location of body, or even body movement. So it's that interdisciplinary nature that makes it really complex, but also makes it quite an exciting subject to teach and to research. And what it demands then is an ability to integrate all these multiple streams of evidence, often in the context of high variability, where we might have incomplete or degraded remains. Another critical point then is that taphonomic processes are highly context specific. And the biggest challenge that we have is that there is not one universal pattern of decomposition. Although there's elements of decomposition that we can predict, if we put bodies in different contexts, they'll behave differently. So a body burrowed in a shallow, acidic woodland soil like we have here in Wales will decompose completely differently to a dry coastal sand dune in Cornwall, which is also in the UK, or even beneath a concrete floor in urban Birmingham. So although the UK is small, we still see those really diverse decomposition environments. And that's because decomposition rates are influenced by an absolute wealth of different factors, both intrinsic to the body and extrinsic. So things like temperature, humidity, soil pH, burial depth, scavengers, body mass, clothing, all of those different things can have really significant impact on how the body changes, and therefore the timelines that we might try to determine once we've recovered those remains. Many of the established decomposition studies that we've heavily relied upon historically have been conducted out of institutions like Tennessee, Texas, or Australia, and they just can't be transported into UK environments. So what it's really important that we do is develop regionally specific taphonomic data, both in our education, our research, and our practice. So students and future practitioners need to be trained on not only recognizing what standard decomposition sequences are, but how to critically assess those and how to think about how local environmental conditions might accelerate or otherwise alter those changes. And then there's other things that we have to think about in terms of the ethics, the cultural and the legal considerations whenever we're handling human remains, and all the issues that we have with body donation and challenges like that. So when we're thinking about how we teach forensic science, it's important that we embed those into the training. They don't just need to know scientifically what's going on, but they need to understand how their work fits in a much wider cultural, societal implication as well. So at Wrexham and at many other institutions in the UK now, forensic taphonomy education is trying to integrate all of these elements. So scientific rigour, interdisciplinary understanding, and critical environmental awareness with ethical responsibility. We need people who are not just able to analyse the dead, but do so in respectful and reflective ways, because that's the nature of forensic casework here. So if we think about the UK landscape more specifically, we've got this complex mixture of challenges and adaptations and exciting innovations that we have to consider. So how we teach our students, how we engage with search doc teams, how we take data from casework and integrate it back in is really important, because these are the things that are going to help us to find missing people, to help us respond to natural disasters, and to help us see results in criminal justice cases. So in the United Kingdom, then, we are often compared to other English-speaking nations like the United States or Australia, but where we operate is uniquely constrained, but we're trying to be creative and involve our research environment. And first and foremost, it's important that we acknowledge the fundamental reality that is that UK currently has no operational human decomposition research facilities. So, unlike in the US, where the University of Tennessee's anthropological research facility started pioneering human taphonomic research more than 40 years ago, or more recently, work that's been done in Canada, Australia, the Netherlands, we've got no equivalent centre here in the UK where we can do human research. But this absence, sorry, it looks like somebody's unmuted their microphone. Okay. This absence in the facility to do this research doesn't reflect the scientific interest or the forensic need. There are lots of practitioners here that want human decomposition research. There are lots of challenges with that, ethically, legally. A lot of it comes from a major legal barrier, which is the Human Tissues Act, which was introduced in 2004. This followed a lot of public scandals involving hospitals that were retaining human tissues and organs that they weren't supposed to. And although it doesn't necessarily make taphonomic research facilities illegal, it makes them really hard to set up, and the governance of such facilities is really unclear. So while the Act was really critical for rebuilding public trust in medical research and healthcare, its stringent regulatory conditions on how we use any form of human remains for research, including in forensic study, make it a real challenge. It's not completely out the question, and there are lots of academics who are trying to move forward in this area. But unfortunately, it doesn't look like it's something that's likely to happen anytime soon. Sorry. Please. Thank you. Additionally, because it's not an established donor process already here in the UK, we'll need to do a lot of work with potential donors and the public sensitivity to make sure that people trust that what we're going to do with these bodies in the future is ethical and has scientific and moral benefits to it. So despite those constraints, then forensic researchers have responded with adaptations and innovation. Like a lot of places, we are doing a lot of research using non-human animal models, such as domestic pigs. They share significant anatomical, physiological, and decomposition patterns with humans, which most people think makes them an acceptable proxy. We do know they're not perfect. They don't have the same diets. They don't have the same diseases or take the same medications as we do. But currently, we don't have many other options. We tend to say that these are controlled and ethically approved studies, but that doesn't mean that they're not without ethical considerations. But they do still allow us to study decomposition sequences, scavenger behaviour, and those things in a fairly scientifically rigorous manner, but not still without significant research costs involved. So there's a lot of research initiatives now distributed across different universities. UCLan, for example, has one of the biggest decomposition research facilities called Traces. There's been research out of Staffordshire and Keele universities, Cranfield as well, particularly around the forensic archaeology side of things, and some more emerging work now at the University of Derby and the University of Kent. So although none of these institutions are working with human remains, collectively they are starting to build a rich UK-specific body of knowledge around decomposition. And that's what we're doing here at Wrexham as well, trying to contribute to this national effort through research that combines different studies using animal sources and technology, essentially. So we're working in areas such as cadaver dog training, drone-assisted search methods, thermal sensing, photogrammetry, all those sorts of things. And one of the big things we're working on at the moment is digital education tools. And one thing that we've launched, which is called Decompanion. And I'll discuss that later later on, but it offers a structured and open access resource for teaching decomposition processes, even when large-scale decomposition facilities and certainly human research facilities aren't available. Okay, so one of the biggest challenges then we need to think about is how we integrate what we're learning in the research facilities into what we're teaching our students in the classroom. And these research innovations are things that work really well in practice, but are hard for students to conceptualize without necessarily going to the research facilities and using the equipment. And we recognize that that's not always practical. They are quite sensitive areas. We don't want to disturb the research too much. They can often be quite inaccessible areas for students that maybe have disabilities, for example. And all of these things we have to kind of build into the way that we teach and research in these areas. We also have to think again, like I said, about the complexities of the microclimatic differences that we see here. So a lot of the work that we need to do is thinking about all the different decomposition environments that we may see, and then how we might be able to use or standardize things across those regions. Even in the UK, we've seen that there's a massive difference in some of the terminology we use and some of the data that we're collecting. So even though I might do a very similar research project here as somebody down in Kent is doing, because we're not necessarily approaching the research in the same way, we can't use that data together to kind of think about a much broader research application than the one we're doing. So we're relying more now on organisations like the British Association of Forensic Anthropology, the Chartered Society of Forensic Science, and networks like the Taphonomy conference to help us to share data and to establish these research networks so that we can look at innovations and best practice. And the growing emphasis is still on those interdisciplinary collaborations, bringing those different practitioners and researchers together. In addition, cautious but important steps are going on in public engagement. So later this year, myself and a colleague will be talking at something called Pint of Science, which is a public science engagement, and we'll talk about decomposition research literally in a pub down the road to hopefully get more people in Wrexham aware of the work that's going on and interested in supporting it going forward. So again, it's about that environmental specificity and that ethical sensitivity as well. So what are the educational innovations that are happening then? Well, one of the biggest and most exciting and promising developments, I think, is the way that we are starting to think about how we teach forensic taphonomy, both at Wrexham and at a number of other universities in the UK. We recognise that we don't always have the space to do the practical work and the research, and we certainly don't have the human remains, but we need to link that scientific knowledge to real-world forensic practice. So at Wrexham, we are very consciously designing our forensic science curriculum to not just be theoretical, but to be reflective and operational. We don't just treat taphonomy as a niche subject that's often reserved for postgraduate anthropology research. We are thinking more now about how we can integrate it across multiple stages of undergraduate and postgraduate study. So from very early on in our undergraduate forensic science program, students are learning about principles of decomposition, environmental influences, and things like that. And they get the opportunity to engage in practical sessions, going out onto our research facility, collecting environmental data, even just helping us to maintain the facility. It requires a lot of weeding and things like that. And all of those sorts of things really help them to think about the challenges of the field work and develop those skills that they'll need to set up their own research projects in the future. But one of the major pedagogical frameworks that enables this applied approach is something called the scale-up model. And that stands for Student-Centered Active Learning Environments with Upside-Down Pedagogies. I appreciate that it's a bit of a mouthful and it's not a term that I came up with. It was originally developed in the United States for STEM education. And basically, it looks at traditional teaching methods, but places students at the center of the learning process. So in a scale-up classroom, the physical space is radically restructured. And you can see on the screen here an example of one of our teaching labs. It looks fairly standard with rows of desks facing a lecturer, but all of these desks are on wheels and can be moved around to put students into small groups working around tables. They're technologically equipped, so students can put their own content up on the screens when needed. There's big whiteboards all around the classroom that students can go and collaborate on as well. So the academic staff member acts more as a facilitator or a guide. So rather than me delivering a 2-hour lecture, there'll have been content that students may work through in advance, reading, short recordings, those sorts of things. And then when they come into the classroom, they're presented with much more complex scenarios. For instance, a missing person presumed to be buried in a woodland environment. The students would work collaboratively to think about what soil changes they might need to see, what insect activity records would be helpful, what's the decomposition timeline, is there a plan for an appropriate search and recovery strategy, what tools that they might need for that. So these sorts of mock crime scene scenarios, which integrate both classroom work, field work, and work that students can do remotely, really helps to mirror the reality of forensic casework. We know forensic investigations rarely come with complete data, linear timelines, straightforward interpretations. So we need students that can do that critical thinking, teamwork, resilience, adaptability, not just memorize facts from historic research. And that scale up model ensures that forensic taphonomy education isn't something that's passive, it's immersive and it's interactive and it's operationally relevant. So it could be things like scattered remains scenarios, clandestine burial excavations, or complex mass fatality simulations where students would start in a classroom, go out into the lab, bring some things back to test. All of these different approaches then allow students to then start to think about the projects that they want to do. And some recent projects we've had have looked at how burial depth affects insect succession, how heavy rainfall impacts surface deposition, and how vegetation regrowth can also help with indicating body deposition sites. Another key thing then is the development of the DCompanion database. And this is really an opportunity that has been led by a colleague of mine called Dr. Paige Tynan. And it was part of her PhD research. And she recognised that there was lots of student-led decomposition happening out there, lots of bits of work done on post-mortem interval, and particularly we had lots of undergraduate and MRes students that were doing valuable work that could contribute to practice, but it wasn't at a stage where it could be presented at a conference or written into a journal article. And they needed a better way to be able to share that research, but also that they were finding it difficult to find that data. So they needed somewhere that they could also access that information. Sorry, I'm just going to go back. I've gone too far. Apologies. There we are. So she created an online tool called DCompanion, which is the open access decomposition database. So she did some systematic documentation of pig decomposition under various environmental conditions. She did lots of data collection. She produced 3D models. And what this now allows students to do, even if they don't physically go to the site, they can look at decomposition sequences and they can access a lot of data about decomposition studies that we were doing in a remote or a blended learning way. And what this allows, if you think back to the pandemic, when we couldn't do any research, we couldn't do any field work, students were restricted. But this accessibility means that students that aren't able to participate in fieldwork or don't feel ready to participate in fieldwork aren't completely missing out. It's still in the early stages of rollout, and we've got some ambitious plans for expanding it, like adding more environmental contexts, adding more thermal and drone imaging data sets, thinking about how we can turn more of the resources into interactive training models for practitioners and students, and also then building in more data sets from other institutions so that it's a more collaborative platform. So some of the key challenges that we're seeing then are kind of led by the fact that we are a small university and we are a small country. So there are definitely challenges with the physical space that we have available to us. So other than the traces facility, most of the decomposition research facilities in the UK are very small. And therefore our sample sizes are limited, and then trying to get published with a very small sample size is problematic. And what we're finding is multiple students and researchers are repeating the same projects because they don't think they've been done before, and it's just because they haven't been published. We also had to be very conscious that we don't repeatedly use the same area for decomposition research. We don't fully understand the long-term impact that decomposition has on soil. So if we use that place for research 5 years ago, how it might affect the research that we do today. We're also limited on spacing, so we often have to put the remains much closer together than we would perhaps like to, and that is problematic. But the main thing is that there are huge amounts of small-scale studies with lots and lots of useful data that could be combined or influence future research that currently nobody has access to. So what we're hoping to do through this platform is synthesize more of that into publications, share it with police forces and forensic recovery teams, and present it on more international fronts, as well as incorporate it into our teaching materials so that what we're teaching forensic students and future researchers is more up to date and more comprehensive than there currently is. And what we need for that then is more people to get involved in decomposition research, and also more people to start posting and integrating on the platform. So although major progress has happened, there are still these challenges that we have to deal with. And funding is always going to be the biggest one. So decomposition research takes a long time and costs a lot of money. So the more we can think about how we reduce those costs, how we minimize lost time on these research facilities as well, and how we think about making best use of that data across various platforms, the better. And the biggest thing then is that gap that still exists between academic research and operational practice. So whilst research is often flourishing, there's a bit of a disconnect between what researchers are doing and what practitioners want. So what we need is more practitioner engagement. So we need forensic search teams, scene managers, DVI experts to be setting our research priorities. We need to hear about more cases where taphonomic research has been used or could be used to address those problems. So by going in a bi-directional dialogue where we can inform the practitioners and they can inform us, we can see that the decomposition research will become more accessible, and also more useful going forward. So there's lots of opportunities to grow. We can think more about those data sets. We can think about improvements for students from more diverse areas. This online database helps us to address a lot of health and safety concerns, a lot of ethical and insurance restrictions, the lack of availability of field sites, and some of those other financial and logistical challenges for students and researchers trying to travel to these areas. But we don't want to remove the hands-on practical approach. We think that that's still vital. the more time that students can spend doing this practical work, the better they'll be set up for the types of roles and the research that they will do in the future. So we're hoping to build more of a collaborative national and international research framework, more partnerships between academia and operational teams, and that will then give us richer taphonomic data sets and make the education in taphonomy more inclusive and more professionally focused. Lots of exciting things to come, I'm sure, with AI, VR, and some of the microbiome research. I think it's a really good time to be getting into taphonomic research. There's challenges, but I think there's lots of opportunities as well. So hopefully I've shown that we have got some innovative educational models. We've been able to research and adapt our research with professional integration so far, but there's more to be done. And then even when we don't have access to large-scale human decomposition research facilities, we can still make meaningful progress. It's a fast-moving area, so there's lots of opportunities, and I'm always keen to speak to more people that are interested in this area and interested in collaboration. So thank you very much, everybody, for your time and attention. I'm happy to take any questions, or if people want to connect with me on LinkedIn or things like that and discuss anything further, either around the education side or around the taphonomic research, then that would be great. Thank you so much.
Right. Thank you so much, Amy. It was very insightful. Thank you so much. So please, any questions for Amy? Any questions for Amy?
I can just see one here. Are there some things besides weather, animals, and environment that can't be identified at first but are important? What other types of things affect human remains? These types of things can affect evidence, making it harder to solve. Yeah, so one of the things that we don't know very much about is how certain diseases and medications affect decomposition. So, for example, if somebody has had chemotherapy, for example, we imagine that that would have significant impact on the insects. You know, the chemo is toxic, so I don't imagine that many insects would want to colonize that body. But particularly where new medications are developing, and even where they do have human decomposition research facilities, the sample sizes that may be available where they have taken certain medications or had certain medical diseases, conditions, are very limited. So we don't really know that much. And again, that's where we can combine lots more smaller data sets. We might be able to start seeing a bigger picture about how those intrinsic factors are affecting decomposition. And then these types of evidence can affect the evidence, making it harder to solve the case. Do you think they somehow help reach a conclusion? I think one of the challenges we have with any taphonomic research is we're never actually going to know if we are 100% right. So we can have theories about time since death, we can have theories about deposition sites, but unless a perpetrator confirms that to us, we're never going to be able to confirm definitively that that is exactly the right case. And often when we're doing the taphonomic research, it's almost too controlled. So we don't allow enough environmental variation and things like that. We control it so much that then it's also not reflective of real casework. So it is a challenge. Is there some tool or technique used for detection or analysis of taphonomy? Lots of different ones, actually. A lot of wet chemistry can be used. So a lot of soil tests can be done to look at changes in the pH. There's a lot more now being done, like I said, with microbiome. So we can use some DNA analysis to detect whether certain microbes are present and how many of those. A lot of research into volatile organic compounds. So that's the scents that come off the body as it decomposes, and trying to work out which of those scent profiles it might be that a cadaver dog is detecting. Specific degrees to pursue forensic taphonomy. A lot of it has been done out of forensic science. But taphonomy itself is more of an archaeological, paleontological process. Anything that has good chemistry or anatomy is going to be helpful, and really it's kind of the overlap between those two areas that we see forensic taphonomy research kind of sitting in. Thank you so much. There were some really good questions.
Okay, thank you so much, Amy, for answering all our questions.
No problem. It was great to speak to you all today. Thank you.
Yeah, we're very grateful. Okay, so without wasting much time, moving on. If you just joined